Hello to all the families and supporters of the 5P minus syndrome community. My name is Megan Leston, and I am the co-founder of the Cri du Chat Research Foundation. I'm also the incredibly proud mom to my son Liam, who is now 11 years old and lives with 5P minus syndrome. And our foundation's mission is to accelerate translational research, focus on developing treatment options for this rare disease. Work that just would not happen without our organization's efforts, our patient community cooperation, and all the different stakeholders in the field dedicated to developing therapies for this rare disorder. Today, we're hosting this recorded webinar to offer our patient community important insights into the drug development process for rare neurodevelopmental disorders. And I would like to introduce our very esteemed guest today, Dr. Yael Weiss, who has served our organization on the periphery as an invaluable mentor for our organization's work. She's a champion for rare disease and rare disease drug development, and she's an advocate for the impact that patient advocacy groups like ours and their communities can have in transforming the landscape of drug development for rare diseases. Her passion for this field led her to establish a company called Mahzi Therapeutics. Yael, thank you so much for being here today and for sharing your expertise with our community. We are honored to have you. And to start, could you share more about your professional background and what led you to become the CEO of Mahzi Therapeutics?
Thank you, Megan, for having me. I'm honored to share my experience with the community. I am the CEO of Mahzi Therapeutics, as you said. I'm a physician scientist by education, but I have transitioned to the pharmaceutical industry many years ago because I find that the interface between science, medicine, and drug development is key to successful development of therapeutics. I started Mahzi 3.5 years ago based on my observation that many patient communities, such as yours, are now advocating for themselves, developing therapies themselves, and really need support and expertise in order to take the therapies into the next stage of development. So many patient advocacy groups have the resources and the ability to fund basic research and to fund proof of concept studies in an academic lab, but most of them do not have the ability to take these findings and take one step further and make a drug out of them, which means testing the safety of the drug and taking it into clinical trials. And this is what we do at Mahzi Therapeutics.
Well, you certainly have a remarkable background, and thank you so much for all the very impactful work you're doing for the rare disease community, especially neurodevelopmental disorders. Before we dive more into the therapeutic modalities for neurogenetic conditions, could you give a brief overview of basic genetic concepts?
Sure, I'm happy to try and explain this in a way that everyone will understand. So each one of us has genetic material that comes from the mother and the father. This genetic material is packed into chromosomes. Each chromosome contains genetic information that is specific to that chromosome, but together we have 46 chromosomes. 23 come from the mom and 23 come from the dad. The chromosomes contain all the information that is needed to make us what we are, to give us the ability to develop properly, to speak, to eat, to drink, to do everything that we do on a daily basis. The genetic material is a code. It's like a computer code, but we're not made of computer code only. This computer code is actually translated into proteins. These proteins are the ones that actually are the functional elements of the body. So these proteins can become tissues. They make up our different tissues. They help build our tissues. They also do daily functions such as cutting different things, getting rid of old proteins and cells in the body. And they also float around and have different functions like carrying things in the blood. Each of you has seen your list of proteins when you go to do a blood test. When you look at the blood test, all the proteins that are listed there have different functions in your body. And the way the doctor determines whether you are healthy or sick is looking at the levels of these proteins. If they are normal, everything is well. But if something is deviated from normal, that means that something is wrong in your body, like in your kidneys or in your liver or in your muscles, etc. So you know proteins, you just don't know that these are the most common and most important things in our body.
What would you say is the basic biology that we're aiming to repair in 5p minus syndrome?
So in different genetic conditions, the code is broken. In some disorders, just one letter in the code is broken. In 5p minus syndrome, a whole chunk of data is missing. So this chunk of data codes for a number of proteins, and now the person that has this missing data is missing the proteins that are actually made from this data. So a patient that has 5p minus syndrome has one set of data and not the other set of data, and one set is not always enough. In some cases, proteins we can do with only 50% of the protein that we have in our body. In other cases, we cannot. So some of the proteins on that chunk of data that's missing from in the 5p region of the chromosome are critical, and we need more than 50%. And this is what we're trying to do to restore that missing information.
Thank you. That was so helpful. So, with that foundation, let's move into the therapeutic landscape. There are different approaches being used to develop genetic medicines for neurodevelopmental disorders, including disease modifying approaches like gene therapies and antisense oligonucleotides, also known as ASOs, along with the use of approaches that are not disease modifying, like small molecules. Can you walk us through what these strategies are and how they are being administered to patients with neurogenetic conditions?
If you don't mind, I'll start with small molecules because these are the medicines that most of us know because we use them on a daily basis. For example, when you have a headache, you take Advil or aspirin if you have heart issues, or any other drug for hypertension or diabetes that all of us know. So these are small chemicals that we take to alleviate symptoms. So if you have a headache, you take Advil and your pain goes away, but you didn't really solve the bottom line. Why do you have the headache? Or if you have diabetes, you take a pill that reduces your glucose, but you're not actually solving the diabetic situation. You still have diabetes. So small molecules are really good to treat symptoms, but they don't, in most cases, they do not fix the underlying cause of the disorder. And this is why we're looking at genetic therapies where we can actually try to solve the underlying cause of the disorder by either replacing what's missing or reducing something if there's too much of it, or trying to restore the function by fixing or editing the missing code that we have in certain conditions.
Can you dive a little bit more into what is a gene therapy?
Gene therapy is a therapy where we restore something that's missing. And the way we do it is by inserting the missing gene into a virus. We all know that viruses infect our body really, really well. We know that from COVID. We know that from many other infectious diseases that are caused by viruses. And we take a virus that doesn't cause symptoms in humans, but does know how to get into our cells. So this virus will not cause any illnesses, but it will get into the brain and go into the cells that need the missing protein. So we take the gene, we package it into the virus, and we let the virus infect the cells in our body and make the protein inside our cells.
So how does that look for an actual patient? How is it administered?
We make the virus in very large containers and we concentrate it into a small syringe. And the syringe is actually used in order to administer the drug into the spine directly. And from the spine, it goes into the brain. So in some cases, we give it into the spine, so on the lower back. And in some cases, we give it right next to the neck so that it gets closer to the brain. And in some cases, we inject it directly to the brain. So it depends on where we need the protein replaced and how much virus we need to give.
And is this anticipated to be something a patient would have to do several times in their lives or one time?
So gene therapy is a one-time therapy because what happens is just like when a virus infects your body, the virus will infect the cells and insert the DNA into the cells, and the DNA stays there. The gene stays there and continues to make protein. So for gene therapy, AAV gene therapy and lentivirus gene therapy, which are the two gene therapies that you will hear about, these are both one-time administrations.
Thank you so much for that. Now, can you talk a little bit more about antisense oligonucleotides?
Yes, I can talk a little bit about antisense. Antisense oligonucleotides are also considered disease modifying therapies. However, they're a little different than the viral gene therapies because they do not have this stable virus that brings them into the body. These are small molecules that mimic the DNA, look very similar to the DNA, and have the ability to bind to the DNA in the nucleus of our cells and either inhibit or upregulate certain genes that they bind to. So since they are small molecules, they do not have a viral vector that carries them around. They're not very stable. So you have to give them every few months because they degrade, and you need to give it another booster of therapy in order to continue the effect. These are given into the spine, usually every 3 to 4 months. And it's probably, if successful, it's a therapy for life that will be given every few months.
Well, all of that was incredibly compelling, and I know that overview is going to be very helpful for our listeners. Some say we're living in a precision neurology renaissance. How would you describe the current state of the therapeutic development for rare neurodevelopmental disorders, and what are some of the key challenges, opportunities, and success stories in this field?
So we are living in a precision neurology era. In the last 10 years, hundreds of new genes have been discovered and associated with neurological disorders. So if once your child was diagnosed with Cri du Chat or autism or seizures, now we actually are able to narrow down the cause of these disorders and enable development of precision therapies, the gene modifying therapies that I mentioned previously. We have seen successes in this field. For example, spinal muscular atrophy, a disorder that is deadly, that children usually do not survive beyond their first years of life, now has two different therapies, an AAV gene therapy and an antisense oligonucleotide therapy, and both enable kids to live longer with a much higher quality and much higher and better quality of life. Other examples are gene therapies that are being developed for Duchenne muscular dystrophy, kids with a muscle weakness that ends up being deadly because their muscles do not function and they end up not being able to breathe. Now with the gene therapies, we can extend their lives, strengthen their muscles, and give them much better quality of life as well.
Would you say these advancements or FDA approvals have paved the way for conditions like 5P minus syndrome in terms of advancing therapeutic development for human use in our disorder?
Yes, there's definitely more understanding of the benefit of these drugs. There are regulatory pathways that are being paved in order for rare disorders to get drugs approved, and better understanding of the regulators of the pros and cons of these types of therapies. And thus, the questions that they ask and their requirements are very focused based on their experience of what they've seen before. What are the potential benefits of the drug, and what are the potential side effects that they know have been observed in previous drug development campaigns?
Sure. Our 5P minus families have been participating in data collection through the use of platforms like Simon Searchlight, Matrix, and Citizen Health. Could you explain the importance of participating in a natural history study, either digital or in person, and what the data is really used for in terms of developing a treatment for our syndrome?
So when we start developing therapies for rare diseases, we do not have the luxury of running clinical trials with thousands of patients because we don't know thousands of patients. We know hundreds of patients in most cases. So our goal is to streamline the clinical development as much as we can. And we do that by running very small trials that are called open label studies. What does open label mean? It means that there's only one group in the study and not a placebo group, which is usually in big indications used to compare the drug to giving nothing to really show that the drug is having benefit. So since we are doing an open label study with no control arm, we need something to compare to. So all these studies that you're participating in, and thank you for doing it because we know it takes time and energy and effort to participate, but it's really important because the output of these studies that you're participating in can potentially serve as a control arm, a virtual control arm, where we can say, this is what a child looks like at any age without treatment, and this is what our treatment is doing, and the regulators will potentially accept this as a comparator versus a placebo control.
Many families are participating in biomarker studies as well by donating blood and urine samples. Can you explain what biomarkers are and how biomarkers for 5p minus syndrome will be used in therapeutic development?
Biomarkers are tools that we use in order to assess response to therapy. So we know that when we treat a genetic neurodevelopmental disorder, we may not be able to see positive effects for months after the treatment because it takes time. It takes time for the neurons to accept the new protein that is made. It takes time for the brain to adjust to the new protein and start functioning normally. So we're looking for a way to measure early on if the drug is effective. We're looking for a surrogate that will tell us, oh, the drug is working, just give it time and you'll see the positive clinical effects. So having a biomarker is really helpful for us to take a sample of blood a few weeks after treatment and a few months after treatment and see if that is changing in a response to therapy, and then giving us hope that over time we will see positive response to the therapy from a clinical perspective as well. It can be EG, it could be something in the blood, it could be something in the CSF, the cerebrospinal fluid, it could be an imaging biomarker, an MRI. So we're looking everywhere to see, to find indicators that the drug is working.
Earlier, we briefly discussed your role as CEO of Mahzi Therapeutics. Would you expand on the value of collaboration between biotech, academia, and patient advocacy groups in advancing drug development for rare diseases?
Of course, I'm happy to. There's no doubt that we cannot develop drugs without walking hand in hand with the patient advocacy groups. Many of the disorders that we are working on and developing therapies for have never had a drug in the clinic before. So actually working with you, with the community, and understanding the disorder better, asking you, what would you like to see improved in your children, gives us an indication of what we should be looking for in a therapeutic approach and what to design, what a clinical trial design would look like, because we want to make sure that we're measuring the things that make a difference for you and are important for you. So if this is the first time that a drug is being developed for your condition, we want to design a clinical trial that will precisely measure the things that make a difference to your community, but also things that are measurable.
Can you share more about how our organization is collaborating with Mahzi to develop a disease-modifying therapy for 5p minus syndrome?
Yes. So when you came to us, Megan, you told us about your Liam's condition and the genes that are missing and the deletion. And we went back and looked at the literature and looked at the gene and looked for ways to make more normal protein. So as I mentioned previously, we have two sets of chromosomes, one from the mom, one from the dad. One has a deletion on it, but one is still functioning normally, and that's called a haploinsufficiency. In cases that there's a haploinsufficiency, we can try to make the normal gene make more protein. So in the normal situation without the therapy, it makes a certain amount of protein. But we know that if we can change the regulation of the gene, we can make more protein from that normal gene. So instead of having only 50% normal protein made from that gene, maybe we can make 60% or 70% or 80%, and that will help restore some of the normal function that is needed. So we have looked at approaches to upregulate, to make more normal protein, and we have found antisense oligonucleotides that could potentially do that.
Well, we're certainly very excited about the collaboration between our organization and Mahzi, and we appreciate the ongoing partnership, and we're really hoping for a significant milestone ahead with the development of that ASO for 5P minus syndrome. What advice would you give to the rare disease community looking to gain interests of industry partners like Mahzi and others to work on our condition?
So, as background, the disorders that have only hundreds or few thousands of patients identified do not get a lot of traction from industry because there isn't really a commercial case for a company to take on a drug that there are not many patients that will be treated. And in the cases of AAV gene therapy, they will be treated only once. So my first and foremost advice is to find more patients, to really launch a campaign in which you will advertise the fact that you have an advocacy group, that you have a group that's organized, a group that is running all these natural history studies and developing a therapy. And this will hopefully motivate more families that are diagnosed to join the foundation, or families that do not have a diagnosis to actually go and get a genetic diagnosis. The moment you have a critical mass of patients on your registry, there will be more interest from pharma because you will have a commercially viable number of patients. You will have all the tools that are ready for pharma to develop a therapy. You have the registry, you have a natural history study, you have an understanding of the disorder, and you have potential therapeutic approaches as well. This will make it more interesting and more attractive for a pharmaceutical partner to join in and join forces with you. Yeah.
Finally, to wrap up, what do you think should give our community the most hope that these therapies will become a reality for individuals living with 5P minus syndrome?
The gained experience over the next few years will give us a better understanding of the extent to which these therapies could make a difference in patients' lives. The more therapies that are being developed, the more acceptance there will be by physicians and by families. And there are studies that will be reading out this year for Angelman syndrome, for Rett syndrome, for Dravet, similar neurodevelopmental disorders. And if these studies read out positively and the clinical data is actually promising and the therapies are making a difference in families' lives, there will be a lot more enthusiasm to develop more and more such therapies in the coming years.
Yael, thank you so much for this insightful discussion. We truly appreciate your time, extensive knowledge, and support for our 5P minus community. We look forward to our continued collaboration and hope to hear from you in the future. Thank you so much.
And thank you, Megan, and good luck to all of you.
Yeah.